I'm a PhD student at the University of Montpellier in France, and I work with the collaboration of 3 different research units, which is the Institut Européen des Membranes, the INRAE UMR-Géo, and the Laboratoire of Technology and Environment. So now a little bit of context. Some areas in the world are facing a severe water stress, and it is increasing by the combination of several factors, such as which are the population growth and also the water demand, and more particularly in the field of agriculture. So to face of this problem, reclaimed wastewater reuse is one of the solutions, and this practice can have several advantages by providing fertilizers in the form of nitrogen, phosphorus, or potassium, but also organic matter for the biological activity of the soil, but also can have some adverse effect by providing undesirable salt, which can disturb in the long term irrigation, the structure of the soil, but also provide exogenous microorganisms. So now let's focus on this last one, exogenous microorganism. These figures show the shared microorganism between treated wastewater and agricultural soil. Here you can see that a lot of microorganisms can be shared by these 2 matrices, but also treated wastewater have their specific community and potentially pathogenic microorganisms. But what about the soil microbial community? Last study showed that this practice, so the irrigation by reclaimed wastewater, can promote microbial diversity and composition modification of the microbe in the soil, but also special loss of microbial community involved in the nitrogen cycle. But the study are limited by the different quality used, so the different treatment processes, but also by the agricultural model, so the plant influence and the frequency and flow rate. So here the question is, how do soil bacterial community structure varying during raw and treated wastewater irrigation in the face of different water quality levels? The objectives were to characterize bacterial community in soil when irrigating with reclaimed water of different qualities. So by evaluate the impact on microbiome structure and also the impact on bacteria associated to nitrogen cycle processes. So for this, we use an experimental platform in Muriel-les-Montpellier. It's a little city close to big city of Montpellier in the south of France. And for the experiment needs, we use different water qualities, so from the raw wastewater after screening. Then we use also a constructed wetland treated wastewater, which allows us to remove the a little bit the concentration of some nutrients, but keep a high diversity and the richness of microbial composition of the water. We also use a membrane bioreactor treated wastewater, which allows us to have a physical barrier for microorganisms, and then a control with tap water with the addition of chemical fertilizers. So we irrigate soil with a drip irrigation system, and we followed different parameters in a water and composite soil. Donc first, we follow the physical chemical parameter by ion chromatography. We did bacterial characterization by 16S rRNA gene sequencing. We measured total bacterial abundance by qPCR on 16S rRNA gene. And then we targeted 3 different functional genes. So the MOA for the nitrification, NRG for the denitrification, and FH for the nitrogen fixation. So I will not go in detail on this table, but this table shows typically we have different level of concentration for our different parameters for fertilizers, organic matter, and microbial loads. And what about the response of overall bacterial community structure in the soil? So this graph shows 3 different groups of bacterial community structure during the irrigation period, with one group for the raw wastewater irrigation, one group for potable water, and one other group with our both treated wastewaters. And nitrogen, organic matter, and biomass are the main drivers of the bacterial community structure. Then, if we focus on the bacterial loads, so first in our water, irrigation water, we have 3 different levels. So the lowest one for potable water, the middle one for our both treated wastewater, and the highest one for the raw wastewater. The response of the soil during the irrigation period show a little increase in soil bacterial abundance in soil irrigated by raw wastewater, but no significant increase in our 3 other irrigated soils. Now, if we focused in the temporal evolution of shared bacteria between soil and water, so first at T0, we found that raw wastewater and soil have the biggest dissimilarity in a bacteria community. Then the middle one, dissimilarity, was for our above treated wastewater, and the best similarity for tap water. And the most impacting result on this analysis is during the irrigation period, the increase in shared bacteria between raw wastewater and soil. Now, I'm go in detail about the functional gene. So first of all, for MOULIA gene, the concentration of nitrifying bacteria in soil just increase in soil irrigated by membrane bioreactor treated wastewater and raw wastewater, and a little bit for potable water due to the fertilizers added, and no increase for a constituent treated wastewater. Then for NARG and IF-H, we found the same results, where an increase in a concentration of denitrifying bacteria and N-fixing bacteria were observed for raw wastewater irrigated soil. So in conclusion, as expected, the parameters that most influence the structure of bacterial community in soil were organic matter and nitrogen compounds, and also the microbial biomass provided in irrigation water. Nevertheless, only soil receiving raw wastewater show an increased bacterial biomass, whereas no impact was measured with treated wastewater or potable water. Then the number of bacteria shared between waters and soil only increased when the reclaimed wastewater contained high load of bacteria, typically in raw wastewater, for raw wastewater irrigation. But there's global change in microbial community has no negative impact on microbial processes, biomass related to nitrogen cycle, because the presence of ammonia can explain our increase in nitrifying bacteria in soil irrigated by raw wastewater and membrane bioreactor treated wastewater in comparison with other water qualities. So I want to thank the financial support of the University of Montpellier for the funding of my PhD. I want to also thank my supervisor, and more specifically Jérôme, who is here, and you for your attention.